Hi guys, and welcome to part 2 of this chapter 11. We got through most of this chapter, especially the action potential section last night in class, and I'll kind of briefly touch over a couple other things I want you to focus on. So just a reminder, when we talked about action potential, these were the steps of action potential and the key players, and this is what you should really focus on. There will be questions about these steps of action potential, the key players, what channels open and close during depolarization, what goes into the cell, what channels open and close during repolarization, and then how we restore the cell back up to resting membrane potential. So you should know really well these steps of action potential, and the workshop for this action potential will hopefully be very helpful. Even if you watch just the workshop video, do the handout. Remember to send all your workshop handouts to the MDSC to get credit for them. And hopefully that workshop will help a little more with this too. So repolarizing the cell will reset all the electrical conditions of an action potential. And after repolarization, specifically the sodium-potassium pump, and you have thousands of them in the axon, are what will restore the ionic conditions of putting sodium outside the cell and potassium inside the cell. And here is the sodium-potassium pump and how it will restore our resting membrane ionic composition. So remember, we always want to have more sodium outside the cell, and we always want to have more potassium inside the cell to restore our resting membrane to resting conditions before we stimulated this axon. And to do that, the sodium-potassium pump pumps 3 sodium out for every 2 potassium it pumps in, and that requires ATP or energy. The threshold and the all or none phenomenon, which we talked about, not all depolarization events will produce an action potential. So this means that sometimes you can stimulate a nerve, but it won't kind of fire, or it won't kind of send off its signal down. And for an axon to fire, to be a complete action potential, depolarization must reach the threshold value or the threshold voltage to trigger the action potential. And at threshold, the membrane will be depolarized by about 15 to 20 millivolts to bring it up to about -50 from our -70 resting. Sodium permeability increases, sodium influx exceeds potassium efflux, and we have a positive feedback cycle. And this positive feedback cycle, and I'm going to put a star next to that. You know why I like to put stars. The positive feedback cycle refers to this idea that once we reach the threshold value, it kind of further enforces this idea of opening up more and more sodium channels so that all the sodium rushes in so we can trigger our action potential peak no matter what. And we call this an all or none response, meaning an action potential will either happen completely when we reach that threshold value, or it does not happen at all. Propagation allows the action potential to be transmitted from the origin down the entire length of the axon toward the terminal ends of the axon. The sodium influx through the voltage gates in one membrane area causes local currents that cause the opening of sodium voltage gates in adjacent areas. So this just means that depolarization moves down the axon. Once initiated, the action potential is self-propagating. Propagation in myelinated axon differs a little bit, and since sodium channels that are closer to the action potential origin are still inactivated, no new action potential is generated there. So what that means is once we generate an action potential in a specific area of the axon, it only flows in one direction, because once that action potential is generated, let's say in this part of the nerve axon, once it passes on to the next, those sodium channels remain closed for a while. So that means that the action potential cannot go back the way it came. It only flows down one direction of the axon, which makes it extremely efficient in sending off a signal through our nervous system. So here is showing the generation of an action potential at resting. Here's the depolarization occurring down the axon. And again, further, we see the hyperpolarization peak. So here's an electrode just generating the inside of the membrane potential at resting during depolarization, and then back at resting again after we went through repolarization and back up. Coding for stimulus intensity, action potentials are alike and are independent of the stimulus intensity. The central nervous system can tell the difference between a weak stimulus and a strong one by the frequency of impulses. And the frequency is the number of impulses or action potentials received per second. And the higher frequencies just mean a stronger stimulus. So here we have a subthreshold stimulus does not generate an action potential. Here's actually a stimulus that will generate an action potential. And the stronger the stimulus, the more frequent action potentials will be generated. The refractory period has to do with the time in which a neuron cannot trigger another action potential. So this is where your voltage-gated sodium channels will be open, so the neuron cannot respond to another stimulus. And there's two types. We have an absolute refractory period, which kind of enforces one-way transmission. This ensures that each action potential is an all or none event. And a relative refractory period, this is kind of where an only exceptionally strong stimulus could generate an action potential. So here is a look at the absolute refractory period. This means that no action potentials can be generated during this time, during this action potential peak, and the relative refractory period where the nerve is kind of getting back into its resting state. Only a very strong stimulus could generate an action potential during this time. Conduction velocity action potentials occur only in axons, and the rate of the action potential propagation, or how fast it can travel, depends on the axon diameter. So a larger diameter fiber has less resistance, so a large diameter axon will have a faster impulse conduction. And the degree of myelination, there's two types of conduction depending on the presence or absence of those myelin sheaths. We can have a continuous conduction or saltatory conduction. Continuous means it's slow and unmyelinated axons, and the saltatory conduction is what we use to describe myelinated axons and how our action potential travels in them. And this occurs about 30 times faster. The myelin sheaths act as an insulator to prevent the leakage of charge, and voltage-gated sodium channels are located at the gaps between myelin. So this means that the action potentials are generated only at those gaps between myelin sheaths. So the electrical signal kind of jumps between gaps, and I'll show you that. So here's the stimulus. In bare plasma membranes, the voltage will decay. In non-myelinated axons, the conduction is slow. But here we have in myelinated axons, conduction is fast as the action potential jumps to kind of find these sodium channels. And it'll jump again to find more sodium channels where depolarization can occur again. So this is saltatory conduction, a Latin root, saltar is to jump. So this is saltatory conduction, it occurs faster in myelinated axons. Multiple sclerosis, it's an autoimmune disease, so your body attacks itself. It primarily affects young adults and often young females as early as their 30s, where the myelin sheaths in the central nervous system are destroyed when your immune system attacks specifically the myelin, and it turns the myelin into a hardened lesion called a sclerosis. And this will slow down the impulse and eventually stop the impulse from happening. So demyelinated axons will increase sodium channels, causing cycles of relapse and remission. There's no cure for it. It's your body kind of destroying itself. Only kind of ways to control it or manage it. So here are symptoms, treatments. Maintaining a high blood levels of vitamin D may reduce risk of development. You might notice visual disturbances, weakness, loss of muscle control, speech disturbances, and incontinence. So the inability to control the urine outflow, all having to do with the muscles and a decrease of nervous system control with those muscles. So conduction velocity, we'll briefly talk about this. Group A fibers are the fastest because they have the largest diameter and they're myelinated. So these will be somatic, sensory, and motor fibers of your skin, skeletal muscles, and joints. And they can transmit signals up to 300 miles per hour, which is so fast. Group B have an intermediate diameter, lightly myelinated, and Group C are the smallest unmyelinated fibers. Impaired action potential impulse propagation can be caused by a number of chemical and physical factors. Cold temperature or continuous pressure can also interrupt blood circulation and delivery of oxygen to your neurons. So this is the idea that your cold fingers get numb or your foot goes to sleep and you feel that prickling sensation. So let's talk a little bit about synapses. The nervous system works as information flows from one neuron to another neuron, and they are connected via synapses or junctions that will mediate the information transfer. And we always call the presynaptic neuron is the neuron that conducts impulses to the synapse, which will be sending the information. And the postsynaptic neuron is the neuron kind of receiving that electrical signal away from the synapse. In the peripheral nervous system, it could be a neuron or it could be a muscle or gland cell. So here's a look at different types of synapses. We have an axosomatic synapse, which is an axon connecting to a soma or a cell body. An axodendritic synapse is an axon connected to a dendrite. And an axoaxonal synapse is an axon connecting to another axon. And here is a great kind of cool picture of those synapses. So here are the different types of synapses. I won't ask you guys too much about different types of synapses. They can be chemical or electrical, and specialized. The chemical synapses are the most common type, and that's because they are specialized for the release and reception of neurotransmitters. And you should know that neurotransmitters are kind of the chemical that's released across the synapse, as we kind of learned about in the neuromuscular junction with acetylcholine. So the transmission across the synaptic cleft is the space. It will prevent nerve impulses from directly passing from one to the other. It's usually a chemical event that depends on the release, diffusion, and receptor binding of a neurotransmitter. And this just ensures the kind of connection between neurons and the unidirectional communication between the neurons. So here are chemical synapses. Again, the specifics of chemical synapses, I won't ask you too much. I focus more on the action potential in this chapter, but you should understand the general idea of this because it goes hand in hand kind of with the muscle and the neuromuscular junction. So the action potential, we'll go through these 6 steps briefly. The action potential arrives at the axon terminal, and this causes voltage-gated calcium channels to open so that calcium enters the axon terminal. And you can see this occurring here. I know it's a little small to see, but the action potential is arriving, opening up the voltage-gated calcium channels, and calcium is entering. Number 2, the voltage-gated calcium channels enter, and that calcium entry is what causes the vesicles to release the neurotransmitter from that presynaptic terminal. And that neurotransmitter is seen here as being released into the synaptic cleft via exocytosis from those vesicles. The neurotransmitter will diffuse across the synaptic cleft and will bind to specific receptors on the postsynaptic membrane. The binding of these neurotransmitters will open ion channels, creating a graded potential, so kind of a localized kind of change in charge on the membrane of the postsynaptic neuron. And you can see here the binding of the neurotransmitter here will cause the opening of a channel to allow ions to pass through, usually sodium entering, to cause a depolarization in that area of the postsynaptic membrane. Neurotransmitter effects will always be terminated. So we're not just constantly throwing out neurotransmitters because we only want the neurotransmitter to bind to cause the continuation of this signal transmission when we want it to. So neurotransmitters will always need to be terminated. They will be reuptaked by astrocytes. They'll be degraded by an enzyme, or they'll just diffuse away from the synaptic cleft so they can't constantly kind of stimulate that postsynaptic membrane. So those are chemical synapses. There might be synaptic delay, which takes time for the neurotransmitter to be released and diffusing across. Synaptic delay is always the rate limiting step of the neural transmission, but it's not very noticeable because it's still so fast. Electrical synapses are less common. I'm going to kind of pass over these a little bit. Postsynaptic potentials refer to the fact that neurotransmitter receptors will cause graded potentials that can vary in strength. And basically, there's just 2 types of postsynaptic potentials. And what these are referring to is basically neurotransmitters can excite the postsynaptic membrane, or they can cause an inhibition response to the postsynaptic membrane. And that's kind of all that we'll talk about here. Here's just a brief comparison of graded and action potentials. Again, we didn't focus a lot on graded potentials. I wanted you to focus on action potentials, but these are kind of good review slides and review charts for those two. Neurotransmitters then. So neurotransmitters are the language of the nervous system. There are 50 or more neurotransmitters that have been identified, and most neurons make 2 or more types of neurotransmitters. And they're usually released at different stimulation frequencies, and they're classified by their chemical structure and their function. And the big one that I want you guys to know is acetylcholine. It's the first identified, it's the best understood, and it's released at the neuromuscular junction. So when the neuron comes in to generate a stimulus to a muscle cell, it's synthesized from acetic acid, and it's always degraded or controlled by the enzyme acetylcholinesterase. I won't ask you guys too many other of the neurotransmitters, but there's lots of different kinds here. Dopamine, norepinephrine, and epinephrine are some of your adrenaline type neurotransmitters. Serotonin, histamine, all of these are widely used in the brain. So they play roles in emotional behaviors and biological clocks. Sometimes there are imbalances with these neurotransmitters associated with mental illness or depression. So the idea that mental illness and depression is not real is totally false, because there are imbalances in the brain between these neurotransmitters in someone who battles depression or another mental illness. So if you do struggle with mental illness, please seek help, because it's definitely a real problem that oftentimes you do not have control over, because there's imbalances with these neurotransmitter or ion concentrations. Amino acids make up all proteins, and amino acids are also neurotransmitters: glutamate, aspartate, glycine, and the gamma aminobutyric acid. Peptides are strings of amino acids that have diverse functions. So again, these are all neurotransmitters that have very diverse functions. And again, neurotransmitters are the language of the nervous system. These are released between synapses, so between neurons that are communicating. So we have lots of different neurotransmitters. So a lot of scientific research and study is spent learning these, because this is how your nervous system communicates. Caffeine, in this example, will block an adenosine receptor. So adenosine is a potent inhibitor in the brain. So that's what caffeine specifically does. Gasotransmitters like nitric oxide, carbon monoxide, and hydrogen sulfide gas. These will directly alter receptors in the brain. Ecanoabinoids are lipid soluble. These are believed to be involved in learning and memory, and they may be involved in neuronal development, controlling appetite, and suppressing nausea. They act as the same receptors as THC, which is the active ingredient in marijuana. So this is why a lot of marijuana is very legal, especially in medical uses, because it helps to suppress and control nausea or control appetite. So often they'll use marijuana or the active ingredient, this neurotransmitter, in patients undergoing chemotherapy, who it's hard for them to eat because they're nauseous all the time, to be able to eat. Functions of your neurotransmitters can be effects or actions. They can have excitatory or inhibitory actions. Just means that acetylcholine, for example, and norepinephrine will bind to at least 2 receptor types, specifically with opposite effects. So depending on what receptor these neurotransmitters will bind to, will affect their kind of the effect. So acetylcholine will excite a skeletal muscle cell, but it will inhibit cardiac muscle cells. Their actions can be direct versus indirect. Again, I do not ask you guys many questions about neurotransmitters, so I'm going to skip over this part, but I include it because it's interesting to read through. G protein linked receptors are involved in transmembrane protein complexes, and they cause wide metabolic changes. And I won't ask you guys questions in this test, but we might see G protein linked receptors come up in a different section. They're muscarinic acetylcholine receptors, and they kind of work to the G protein signaling mechanism is kind of like a molecular relay race, where a ligand is the first messenger, where the neurotransmitter binds and activates the receptor. The receptor activates the G protein. The G protein activates what we call adenylate cyclase. And then adenylate cyclase converts ATP to cyclic AMP, which is CAMP. And then CAMP will then change the membrane permeability by opening or closing other channels, or activating other enzymes, or activating other specific genes. So this is a G protein linked receptor. You don't have to know all the steps to this, but just know it's kind of like this relay mechanism where one thing activates something else, where the end product, this is cyclic AMP or CAMP, can have several different effects throughout the cell. Neural integration is neurons functioning together in groups. Neuronal pool is the functional groups of neurons. And again, I won't ask you guys too much about this, but your neurons sometimes are working together in groups or pools to process from one pathway to the next. There might be one question about reflexes, and reflexes are just the rapid automatic response to stimuli. The particular stimulus can always cause the same response, and it occurs over pathways called reflex arcs that have these 5 components of a receptor, a sensory neuron, a CNS integration center, which is usually the brain or spinal cord, and then the motor neuron and the effect. So there might be one question about reflexes, and that is where we'll end this chapter. We'll see you guys Thursday night as we start chapter 12, and I hope you guys are all doing well. Take care.